All right, welcome to our first lesson from the year. And so the idea of this lesson is just get us reacquainted with the experimental design process. I know a lot of it will probably be a little bit of a review for everybody, but it's going to be really important because especially on the exam and for our class in general, being able to design a well-controlled experiment is going to be your number one skill. It's going to be super important, not just for what they expect you to do for questions on the AP exam, but also what I'm going to expect you to be able to do for all of our labs that we have in class. So it's a good place to start. So AP Bio is really big on what's called scientific inquiry, or the way in which scientists explore the natural world around them. How do they ask questions about observations that they have, and how do they get to a satisfying conclusion of those observations in a methodical way, right? In a way in which they can be confident about their conclusions, or at least they can be realistic about the error that might be present in their conclusions. So first, we want to talk a little bit about how the scientific inquiry process works, or the scientific methods. And you might see here on the left some different steps of the scientific method that you've been taught and that you've gone through probably a number of different times in your career as a student at this point. And the problem with those is that it doesn't necessarily reflect the complexity that exists in actual scientific inquiry and actual scientific research, and how interrelated all of these different steps are and how non-linear they are. So, for example, we don't always start the inquiry process by making observations, right? And even if we do, it doesn't necessarily flow in a nice straight linear order right through the steps all the way to the bottom. If you can see on the graphic on the right, sure, there are all of these portions that are in the traditional scientific method on the left present, but they're much more fluid. They're interlaced in a much more web-like manner, going from one to the other. Um, so, for example, you might start some scientific research by gathering data and interpreting the data. And then from there, you might realize that there are things within that data that might not necessarily represent the cutting edge of scientific knowledge at that point. So you might need to go to this community analysis and feedback section to get expert ideas from others that are researching the same thing. And from reading some of that literature, you might then have an idea for an experiment. And so you might come over here to the explore and discovery phase, and that might lead you to an experiment where you can gather your own data, right? So it's not a linear type of step-by-step process as is often taught. And when you get into the real world of actually doing scientific research, it is a lot more complicated, and each of these steps or portions might last years, if not longer, to complete. So that's something that we should always keep in mind, even though experiments of that length usually aren't feasible within our class for obvious reasons. So one of the key, most important parts is making inferences and observations. That's kind of how we get an idea as scientists as to what problems need to be solved, what questions we have. And as most basic, an observation is just something we notice from our senses or from measuring tools like meter sticks or pH meters, or all of these things just serve to heighten or make more precise our own senses. From our observations, we make what are called inferences, what are interpretations of what those observations mean based on our prior knowledge. So, for example, in this picture here, we've got a tree, a broken branch hit in the water. We might observe that there is a broken branch on this tree and there's a child sitting in the water, but an inference might be that, oh, maybe he was sitting on top of this branch, it broke and he fell in the water. A different inference was maybe he's playing in the water, this branch broke, it's about to hit him, right? Both of those are inferences, different inferences that we can make from the same observations, right? And so scientifically, we use data, facts, figures, and other evidence gathered, and we analyze that data, especially if it's numeric using statistical tests, to make sure that our inferences are the most plausible for the data that has been given. In linking our observations and inferences, a lot of the times we make what's called a hypothesis or a proposed explanation based on the evidence we have at that point. What's most important about a hypothesis is it has to be something that we're able to test. There has to be a way that we can answer it, right? At the same time, it can't be too broad, right? A hypothesis that we make needs to be uh, narrow enough for one experiment, right? So if I make a hypothesis that's too broad, one individual experiment isn't going to be able to test that hypothesis. Maybe a number of experiments would, but when we're designing an experiment and we're creating a hypothesis for that experiment that we're going to do, it needs to be specific enough of exactly what we're going to test and that we are able to say yes or no, supported or not, based on how we design our experiment. Now, I know a lot of the times in the past, you've been trained to write hypotheses as an if-then statement. And really, the reason you've been asked to do that is so that um, a teacher can really clearly see that you've been able to identify your independent and your dependent variables. And we'll go back over those in a second. But if you see examples like I've linked here or typed out here below from actual research papers, hypotheses in actual research papers are very seldom in an if-then statement, right? What's really important is when you're writing a hypothesis that you're able to write down how your independent and your dependent variables are proposed to be or not to be correlated. And you can see those that is true for each of these three that are written right here. So next, we're going to talk about the experiment part or the procedures that we're going to use to test our hypothesis, answer our question, or solve our problem. So after we have a hypothesis, we want to think about what kind of data we will need to collect in order to test that hypothesis. And then we're going to need to design a set of steps, a procedure that will uh, collect that type of data. Now, in every single experiment that you ever design for our class, they your experimental design will always need to include two groups, at minimum two groups, a control group, so a group within that experiment that uh, you do not subject the independent variable to, and at least one experimental group that you do subject the experimental variable to. So if you were designing an experiment to test the effect of some the varying, let's say maybe you're going to test the varying intensities of light on the growth of a plant. Okay. So you are going to have at least one control group. And the control group would be one that's not receiving the independent variable. And in this case, that variable, what are you testing? Light. So that control group would not receive light. Maybe you put it in a drawer or in a dark room. That's going to be your control group. That's what we're going to be able to compare all the others to. Then our experimental groups, we might have different plants in separate pots that we then put under light of different intensities. And then at the end, compare how much they've grown compared to one another and compared to the control group. So all of your experiments need to have a control group for comparison and at least one experimental group. You could have 4, 5, 6, 8, 20 experimental groups potentially, but you need to have at least one of each. So as we were talking about, an independent variable is a factor that you want to test or manipulate. In my last example with the plants, that was light intensity. The intensity of light is what I'm trying to test to see if it affects what we call my dependent variable. This is the factor that we're going to observe or measure during the experiment, the effect. So the cause, the independent variable, was light intensity. And what do we want to see it does? Well, we want to see if it affects the plant growth. So the plant growth may be measured by the plant height is what I would measure. So when we're designing an experiment, always at least one control group, always at least one experimental group. In our experiments, always only one independent variable, always only one dependent variable. Now, everything else, all of the other factors that we are going to try to keep the same between the members of our control and experimental group, that's going to be called control variables. The other aspects of our experiment that we want to prevent from affecting our dependent variable. So, for example, with our plant example, we only want the light intensity to affect the plant growth or to see if it affects the plant growth. That means the amount of water the plants receive, the type of soil that they're in, the type of pot they're in, even the type of plant themselves needs to be exactly the same for each plant in the experimental group or groups and in the control group or groups. That way, there's no way that we could confuse the effect the soil might have on plant growth and the effect the light might have on plant growth. We're only testing one thing at a time. And so we might and usually will have dozens of control variables. Now, in reality, it's extremely impossible. It's extremely difficult to eliminate the effect of all control variables, especially when we're doing things where our experimental or our subjects are like human beings, but we can do the best that we can. So, for example, we might have an experimental question where we're wondering if taking a weight loss supplement leads to weight loss. Okay. So in this experiment, we would know that our independent variable is the weight loss supplement. We are going to control whether we take or our patients take or our experimental subjects take the weight loss medication. This is what we want to see if it has an effect on weight loss. And what we're interested in, what we're measuring, the dependent variable is weight loss. We don't know whether we will lose weight until we take the supplement. It is what we're going to measure. So in this very simple experimental question, we can easily identify our independent and our dependent variables. So in short, our control group then will be the one that does not receive our independent variable. It's not tested. And our experimental groups do receive, to some extent, the experimental variable. Now, in class, we're actually going to talk about uh, the fact that there is more than one type of control. There are positive controls and negative controls. We're going to do an activity in class about this specifically. But if you would like to and have a copy of this presentation, you can actually click on these links to videos we'll watch in class as well. So a controlled experiment, which is what we're always going to strive to create at AP Bio, is an experiment in which all the variables except our independent variable remain the same, are controlled. So only one variable, our independent one, is allowed to change. So an experiment that is designed to compare an experimental group with a control group is called a controlled experiment. And so ideally, a control group and an experimental group differ in only the presence in your experimental group or absence in the control group of your independent variable, so that your control group can serve as a comparison that you can measure the effect of the uh, independent variable in your experimental group against. So for a quick check, think about this. A drug manufacturer is testing a new drug to treat gout. An investigator dispenses a placebo, a sugar pill that contains no drug, to one group, and the other group receives pills containing the actual drug. Think about in this situation which group is the control group and which group is the experimental group. Now, hopefully, you identified the group that received the sugar pill as the control group. They're not receiving our drug, and so they would be the baseline that we can compare our experimental group, the group that did receive the gout medication, against. So, as we were talking about, the other type of uh, variables we have are controlled or constant variables. You might hear them called constant variables or controlled variables. These are just the ones that we don't want to have change throughout the duration of the experiment so that we can rest assured that they will not potentially have an impact on what we're measuring. So in this example, this ball being dropped from different heights, we might use the exact same ball every single time. So we know that the type of ball doesn't have an effect on how high it's going to bounce. So once we have completed and designed our experiment, we've ran our experiment, we've collected the data that we need. Next, we need to interpret it. Next, you need to interpret it. So after you gather your experimental data, we might create data tables or graphs to represent the data because as human beings, we are visual creatures. And seeing trends and patterns in data is much more easily done visually for us than just having a big table of numbers. That's much more difficult for us to find meaningful trends. We might perform calculations that will determine what results mean. So, like figuring out the averages, percent statistics, and we might identify possible sources of error that could introduce ambiguity into our results. So all of these things are part of interpreting, making meaning out of data. So after we've had an opportunity to do those results, we need to go back and we need to think about how does our data and our interpretation of our data address our hypothesis? So let's draw a conclusion, an answer to our experiment. So after you've organized and interpreted your data, you must draw some sort of a conclusion. You need to decide if your data supports or fails to support your original hypothesis. And so this picture is really interesting because it shows how, depending on how you choose to analyze your data, directly affects the conclusions you might reach. So in this picture, this graph, it says 29 research teams were given the exact same set of soccer data and asked to determine if referees were more likely to give red cards to darker skinned players. Each team of researchers used a different statistical method, so a different way to make meaning out of that data. And each found a different relationship between skin color and red cards. So here on this data, each of these circles represents a different research team. And if this data lies on this bottom line, they were equally likely to give a red card to a player regardless of their skin color. If the data lie on this line here, there's twice as likely to give a red card to a darker skinned individual than a lighter skinned, and the line above is three times as likely. Now, all the green dots represent research teams that from their data analysis, their results showed a statistically significant difference, or darker skinned players were statistically more significantly likely to receive red cards than lighter skinned players. And the gray dots mean that there wasn't any statistical significance to the results. So you can see all of these different groups received the same data. Most of them saw different results, all based on the statistical method they used. And so one of the things that these um, groups would then be subject to after these findings maybe were published would be other research teams arguing for or against the appropriateness of the certain of the statistical methods that they used, right? And that's just a natural part of science. So you can see how just because two groups or more than two groups receive the same data, based on how they analyze that data, can mean very different conclusions. Here is another set of data that compares the number of people killed by venomous spiders in different years, the black line, with the number of letters in the winning word of the Scripps National Spelling Bee. And you can see how this data, red and black sets, are what we call correlated. One goes up, the other goes up, one goes down, the other goes down. Their curve shapes are very similar. So this is what we call correlation, right? But it would be very premature to say that one of them affects or causes the other, right? So we always need to be wary of that. We can see two things are correlated, go up when the other go up, go down when the other go down, but that doesn't mean that that effect is caused by an interrelationship between the two at all. And that's always something we need to keep track of. Finally, scientists communicate their results. They communicate their finding to other scientists, to the public, and this contributes to the growth of the body of scientific knowledge. And communicating it in journals like this allow other scientists to look at and to question and to retest and to object to things that you did in your experiment. And that just improves the quality of what we receive as a final result and also make sure that there are other avenues that can be explored. So, getting down kind of lastly, I want to make sure that we're aware of the difference between two key terms. So, a scientific theory is a well-tested concept that explains a wide range of observations. And a theory is based on thousands of experiments done by, or many different experiments done by different scientists. And it is much broader in scope than a hypothesis. It's far less specific. And they only become widely accepted in science if they are supported by an accumulation of extensive and varied evidence over time. Now, even if something becomes a very well accepted theory, it never can be proven, right? Only things in mathematics really can be proven for sure, certain, because all that is needed is new data or one piece of new information that contradicts the theory to cause it to crumble and go back and need to be reevaluated or redone. So one of the things that we're going to look at in class is a way of writing called claim evidence and reasoning. And this is a way in which you can very methodically, very concisely uh, state a conclusion in a way where your thoughts, the relationship of your data to your hypothesis are all very succinctly communicated in an understandable way because a lot of times students have a lot of problems doing this. And it's a good way to model the writing process so that you're sure that you're including the correct things when you're writing something like a conclusion. So, for example, if you were going to write a conclusion to your data, you would start with a claim. So, a claim would be, for example, how does your data answer your original question? How does your data support or not support your hypothesis? Right? It should be really to the point with no unnecessary words and gets rid of any kind of vague notions. So, what are you claiming after your experiment is the relationship between your results and your hypothesis, your independent and your dependent variable? So, examples might be some of these. All are succinct, to the point, state how an independent variable, smoking cigarettes, might affect a dependent variable, lung cancer, and should be, kind of exactly what you had written down for a hypothesis. Next, you'll use your scientific data to support that claim. All right. So now you are going to go to the data collected in your experiment and cite that as supporting your claim that you're making. Okay. So evidence must be sufficient and relevant to your claim. So not all data is going to be considered evidence. You might have collected data in your experiment that doesn't serve really any purpose as far as evidence at all. And it may be data that you generated for lab, or it may come from a literary source, research that's been done before. And usually the more evidence you have, the better you're able to prove quote unquote or support your claim. And so here might be an example of evidence that you would cite in a conclusion of CER, for example. Right. And here's that same piece of evidence again. And lastly, you have your reasoning. And so your reasoning is an explanation that connects your claim and the evidence. So how does or why does this evidence that you're citing, what's the reasoning? What's the rationale that you're using to say that it is supporting your claim? So why the data that you chose counts as evidence, and why does it support your claim? And acts kind of as a link, a connection for your conclusion. So it shows a detailed understanding of the scientific principles involved and uses correct scientific vocabulary. It might be a few sentences in length, it might be a paragraph, it depends on how nuanced your data is. And it might include a discussion of why you used a specific statistical technique on quantitative data, for example. Okay. So we're obviously going to be going over a lot of this as we go through the year in class, right? We're not going to have a separate test specifically on this Unit 0 experimental design stuff, but it is things they do represent the information in here, ideas that you will be expected to know and be able to use in lab and on tests for the remainder of the year.